understand. Good. They missed the bad part. Um, what we thought was most important to understand is the building block of the capsid. And so we used a combination of X-ray crystallography and NMR spectroscopy to understand the basic building block at the protein level of the capsid. And so this is the CA protein. We found that two copies of it come together. So it's what we call a dimer. And we always thought of it as looking something like the head of a rabbit. So one of the domains forms the head of the rabbit, and then the other domain, and there are two of them because it's a dimer, come together to form the ears of the rabbit. And I'll get back to that in just a moment. So to give you a sense of scale, about 750 copies of that CA dimer have to come together to form the conical capsid. And a cone is an unusual biological assembly, and so we were really curious, how does a protein make a cone? And that's what I'm going to talk about next. So I'm only going to show one experiment, but as a scientist, I know many of you will know this. We spend most of our time trying to figure out why our last experiment didn't work the way we thought that it should. But once in a while, there's a breakthrough, and it's clearly an advance. And this is an experiment that was done by a very talented graduate student in the lab, Barbie Ganser-Pornillis. And what Barbie discovered is she took the pure protein that I just showed you, and she raised the concentration and raised the salt, then it would spontaneously form cones. And so that was an exciting day in the lab. And in those days, we used to collect all of our data on film. And so my contribution was to put my thumbprint on her nicest picture of cones that she ever took. And if you looked at the cones that she was making, so these are again electron micrographs, but now we've sectioned them so you can see through them. On the right, you can see a virus, and so it's quite a complicated structure. It has a membrane around it. It has the central conical capsid. The capsid is dark because it's filled with RNA, genetic material. On the left, on the same scale, is the cone that one of the cones that Barbie was making from just a single pure protein, the CA protein. And what you can see is that they're clearly the same object. And if you look at a cone, it not only has a body, but it also, when we looked at these cones, it was clear that they were closed at the end. So a cone has a wide end, and that was closed, and it has a narrow end, and that was closed. And so it forced us to start to think about how can a protein assemble into a cone and have the cone be closed at both the narrow and the wide ends. And because of Barbie's work, we also had a tool to start looking at that. And I won't take you through that except to say that we ended up deciding that the assemblies, the conical assemblies, belong to a family of proteins called fullerene, architectures called fullerene cones. And so this is a picture of a fullerene cone. And if you look at the body of the cone, then you can see it's composed of six-membered rings. And each of the six-membered rings has around it six more six-membered rings. So it has the exact same symmetry as a honeycomb. And then if you look at the top of the cone, you can see that it has, in yellow, pentamer defects. And so from Euler's mathematical theorem, it turns out that in order to enclose what we call a hexagonal lattice, you have to introduce pentagonal defects. And you have to introduce a total of 12 of them. And in a fullerene cone, there are 7 at the wide end and 5 at the narrow end. And we sort of know that intuitively because if we look at a traditional soccer ball, then you can think of it as the same kind of a hexagonal lattice. So you have white panels that have 6 sides, but it's allowed to close into a sphere because it has exactly 12 black panels, which are pentagons. And that turns out to be universally true, and it's true for the fullerene cones as well. So over the next decade or so, a number of labs, including ours, filled in the molecular details of the capsid. And so this was particularly driven by Owen Pornillis and Barbie Ganser-Pornillis. And so now what you're looking at is a space-filling model where we know the positions of all of the atoms in the fullerene cone. You can see it still has 6-membered rings, but now they're rings made of protein, and we know exactly where their atoms are. They have 6 nearest neighbors. And it turns out that the head of the rabbit that I showed you, it's the dimerization domain that holds one hexamer together with another hexamer. So that's how that building block, and then the ears of the rabbit make these 6-membered rings. So that turns out to be quite important advance because first of all, it shows that the structure was right. But second of all, it shows companies like Gilead Sciences can now do something called structure-based drug design. So now they have a three-dimensional structure of the object. You can look at a hexamer using X-ray crystallography or electron microscopy. You can bind inhibitors to it and ask, they don't bind very well. How can I do medicinal chemistry to make them better and better? And that's called structure-based drug design. And Gilead did a lot of that to make a very potent drug, Lenacapavir. So just to finish this, HIV has been a testing ground for new technologies because it's such an important problem. And this is a paper from John Briggs and Hans-Georg Kreuzlich's lab, in which they're using a technique called cryo-EM tomography. And they're actually looking at 4 different virus particles. So now they have the resolution to actually look at the capsid in situ, inside the virus. And you're slabbing through the viruses. And what you'll see is that they have sufficient resolution to actually render the positions of the hexamers and the pentamers. And so inside authentic capsids, they really are fullerene cones. If you look at the upper left one, it has 7 pentamers on the wide end, and it has 5 pentamers on the narrow end. But one of the things we now realize is that the virus is able to do this in many different ways. And so you can see that it's not just one structure, it's a whole family of structures. So I hope you agree that those are really beautiful structures, and I'm not the only person who thinks that. There are several artists that I know who have used HIV capsids in their artwork, and this is one example. John Walter is a multimedia artist. He had a show that was traveling throughout Great Britain a few years ago called Capsid. It's multiple rooms and it's multimedia, but you can see that the central piece is this something like 15 by 25 foot piece, and right in the middle of it is the HIV capsid. And I think it's attractive to artists because each of the hexamers, although it's clearly a very regular structure, is actually in a slightly different environment. And also, of course, because it represents an enormous amount of human suffering, but also perhaps the hope that we can at some point overcome this disease. So now I'm going to step back and talk to you about what the capsid actually does for the virus. And so on the right-hand side of the panel, I'm not going to take you through that because that's the video that I showed you. That's how the virus produces a new particle. But if you look at the top, once the virus has created an infectious particle, then that particle has to infect a new cell, a T cell, and it does that by recognizing the receptors on the T cell called CD4 and the co-receptor, and that releases the core particle, the capsid, into the cytoplasm of the newly infected cell. And then it can move on microtubules, they're the highway of the cell, to the nuclear pore. And we now realize, it's only just the last few years we've come to realize that it actually can move right through the nuclear pore. It's a very tight squeeze, but it can make it all the way into the nucleus. And during those steps, so the sort of most famous step in retrovirology is taking place, which is called reverse transcription. And so the light blue on the top is RNA. The virus is an RNA virus, but it gets copied into double-stranded DNA by the time it gets into the nucleus. And so then that double-stranded DNA breaches the capsid. We call that uncoating, and it's integrated with the help of the viral integrase into the target chromosome. And this is the reason it's so hard to cure people of HIV, because you not only have to get rid of all the viral particles, but you also have to get rid of all of the T cells that were ever infected, because the virus is archiving a copy of itself in every infected cell. So another experiment that Ute von Schwedler, a postdoc in our lab, did that turned out to be more important than I think we thought at the time, was that she made mutations in the CA protein that makes the capsid. And these are small mutations that weren't designed to disrupt things. And she asked, if I build them back into viruses, does the virus care? And she made a total of 50 of them. And the surprise was that 46 of them were down at least twofold in infectivity. And moreover, more than half of them were down at least 20-fold in infectivity. And so what that said is the virus really cares about the capsid. And if you make even small changes, you'll hurt infectivity. And moreover, she had interesting phenotypes for her different mutants. So some were blocked at assembly, and that's because the CA protein helps the virus to assemble. Some of them were blocked in making conical capsids, and that's because the capsid is made by the CA protein. And those were almost invariably blocked in reverse transcription. So that says that replication takes place inside this conical capsid. Some of them were blocked at what we now understand to be docking at the nuclear pore. And some of them couldn't make it through the nuclear pore. It's a very tight squeeze. And so if you look at the drug lenacapavir, its most potent step that it blocks is entry of the virus into the nucleus and immediately post-nuclear steps. But it also blocks at slightly higher concentrations, the process of assembly and the process of maturation, and then at even higher concentrations, the process of reverse transcription. And so part of the reason the drug is so effective is that it blocks many different stages of the viral life cycle, and that's because the capsid functions at many different stages of the viral life cycle. So I want to talk a little bit about the drug itself. This is a slide that I borrowed from Thomas Silar, who's the Senior Vice President of Virology at Gilead. And after Uta published her paper, they contacted us and said, do you think capsid would be a good drug target? And when a leading drug company asks you that question, you say yes. But we really did think it would be a good drug target because her mutations said that the virus really cared about it. So we helped them to set up a drug screening program, but they're the people who did all of the drug creation and development. That started in 2006. After about 4 years, they decided that their lead compounds weren't useful, and so they started over and made a new set of scaffolds, and that was successful. And so if you look at the graph, what you're looking at is a measure of how much drug it takes to inhibit viral replication by 50%. And you start early on in the upper left-hand corner. The drugs are not very good. They're not very potent, but you do medicinal chemistry and they get better and better. And this was really a heroic piece of work led by John Link and his team. And you can see that they, over the course of the next 6 years, made more than 4,000 compounds. And they got, this is a log scale, so they got about 10 to the 6th more potent until they ended up with the molecule that we know as lenacapavir. And for those of you who are chemists, you'll notice that it's quite a complex molecule. It's very large for a drug molecule, so it sort of breaks the rules of how big a drug should be. And it has stereocenters. It has a total of 10 fluorines, which is very unusual. And so it's really sort of a beautiful piece of modern medicinal chemistry. So this is just to finish and tell you where we currently are. It's now possible to make pure core particles, pure capsids from viruses. And that's shown in the uppermost panels. And you can see they have this nice rounded fullerene cone structure. But if we add the drug, what happens is they become very flat and rigid and angular. And so you can see that in the lower panels where we're adding lenacapavir to the purified core particles. And if you analyze that, what you discover is that what lenacapavir is, this is work from Owen Pornos, what you find is that the drug forces the hexamers into a preferred bite angle. And so you can envision that in a cone, you have to have all different kinds of angles between hexamers to create the curvature. And the drug doesn't allow that. And so it creates tension in the lattice, and that causes the lattice to flatten where the drug binds. And it induces strain, and then the drug causes the capsid to crack. And it also causes it to lose the regions of maximal curvature, the places where those pentamers were. And so that means that it sort of destroys the capsid, and that's why the drug works. So as I said, I had a lab for 34 years working on HIV, and so I thought I would make a few observations. I feel very privileged to have been part of going from sort of very basic research. We just wanted to understand how does a protein make a cone to get to watch through the process of a new drug that looks like it has great potential. And so one point I want to make is that lenacapavir really is an exceptional drug. So it's very potent, that's important, but more importantly, it's long lasting. And the medicinal chemistry that went into that is really very impressive. And it's actually changing how people think about drugs for chronic conditions, because you can envision that you would like to have a slow release, long lasting drug for many chronic conditions. I think it's also true that capsids can make good drug targets, and we and I think others didn't think that when we started the work. So it looks like a capsid looks very stable, and it looks like you should have to bind a lot of drug to it. But in fact, because the capsid functions at so many steps in the viral life cycle, and also because it has to both assemble and disassemble, it has a very narrow energy window over which it's stable. And so if you can perturb it, the virus cares a lot about that. And I think that's going to turn out to be true for other viruses. And then just to finish with a couple of policy observations. So I've talked about the role of academia in our lab, but also other labs. And the point I want to make is that it took us more than a decade to do the work that I showed you in the beginning. We're still working on the problem. And that simply wouldn't have been possible without reliable funding from NIH. So NIH funding was competitive between 10 and 20% of the grants that are go in, get funded. So most grants didn't go in, but we still had the sense that if we kept doing good work and could explain why it was good work, that we would be funded. And that enabled us and others to do long-term research on a basic problem. And that was critically important. And that's important to point out now when the NIH funding is under such a threat. You won't be surprised that I'm a fan of basic research. I think if you look at transformative medicines, so for example, the immunotherapies against cancer, essentially all of gene therapy, the new weight loss drugs, they all have deep roots in basic science. And they come from people studying things like, why don't Gila monsters have to eat for 6 months after a meal? What's in their venom? How do bacteria protect themselves against phage? Problems that we would never have guessed would end up transforming a field of medicine. And so basic science really does matter. I also talked about the role of industry. I'm a believer that most drugs should be developed in industry because of their resources and their capabilities. I haven't talked about the importance of clinical contributions, but caregivers and clinical researchers and clinicians obviously made a huge contribution in this going from bench to bedside to community. Regulatory folks made important contributions and community groups made very important contributions. So for example, community groups in Africa really pushed Gilead to test the drug in pregnant women and lactating women and also in adolescent girls. They all have their own complications for a drug company, but as a result, those groups of people who are really key people, they're people we want to keep from being infected, are now all eligible for the drug. And then finally, I want to finish by saying that, we're sort of at a precipice where the drug is now rolling out and it's going to have to be on a larger scale than it is currently being rolled out. And that's going to take exceptional talent and commitment and to distribute it broadly to the people who need it. And I think that's particularly important, again, because as we'll hear, groups like PEPFAR and USAID, who have been major factors in delivering drugs to the developing world, are currently under threat. And so it will truly be a human tragedy if we're not able to accomplish that. So I'll just finish by thanking the people in yellow are the people who were involved in the early work that I talked about. The people in red are the people who are working in our lab now on current problems like how does lenacapavir crack the capsid. We've been fortunate to have this interaction with the groups at Gilead, including Thomas Seelar and John Link and Steve Yant and their whole multidisciplinary group. That's a picture of the University of Utah Health Sciences Center. That's where our lab is. That's my house. And so I'm privileged that I get to walk up over a hill every morning and I see the Wasatch Mountains in front of me, and I find that quite inspiring, and I find all of these people very inspiring. So thanks to them for their hard work, and thanks to you for your attention.
Thank you, Dr. Sundquist. And I invite you to go ahead and sit on the panel and we'll start filling up those seats. So next up, we have Saba, who is going to really ground what lenacapavir means for patients. As a clinician, she has expertise in actually delivering PrEP. And so we're really excited to hear from her on what the reality is for patients and how this drug can really push forward options in our efforts to combat HIV. So here you go. You want to see from here?
Hi, my name is Saba. I'm a PA. It's going to be hard to top Dr. Wesley's, top time 100 most influential person, but I'm going to be briefly speaking about my experience with the medication Dr. Wesley was talking about, Lenacapavir. So I've been working at an STI clinic for about 4 years. For those who are not aware, there is something called PrEP for HIV. It stands for pre-exposure prophylaxis. It's you take this medication in order to prevent getting HIV. So we do a lot of that medication management in my clinic. And in the past 5 months, we now have lenacapavir for HIV prevention. It's under the brand name Yestugo. So we do have the Yestugo available for patients, and it's been a game changer because traditionally PrEP has been a daily pill. And like Dr. Wesley mentioned, it can be a pill burden for patients to take a pill every single day for something they don't have. So this is something you take every day to prevent HIV. So this has been a game changer. You come into the clinic, you get an injection on each side of your abdomen, and it's good to go for about 6 months. So you have 6 months of protection against HIV. And usually the people who are getting on PrEP medication are at high risk for HIV acquisition, and it's usually through sexual encounters. This medication is readily available to anyone, but usually our patient population will be younger. They don't have a lot of other health conditions. They don't have a lot of other health comorbidities. So for a lot of them, it is the only pill they're on. That's why it can be hard for them to remember to take something every day. They are usually not taking any other medications, maybe a supplement here and there. And one of the barriers or one of the things that it makes it hard for patients to access this medication in the States will be something like insurance. If your insurance doesn't approve it, it's very hard to get the medication. You might be able to fill out an assistance form, but that's not guaranteed. And at our pharmacy, the out of pocket cost for just one dose is about $14,000. So no one's paying for this medication out of pocket. If your insurance approves of it, then you can get the medication. Um, and then in our clinic, the clinic that I work in is going to be a little bit different because we don't accept insurances for a lot of the services we do. We only bill on the pharmaceutical end. So a patient might be able to get this if they're going to primary care where they have better understanding of insurances, because we don't have anyone on our team that deals with insurances because we don't do medical billing for insurance. And then there is still a lot of stigma around HIV. And because there's a lot of stigma around HIV, there is still a lot of stigma around preventing HIV. So patients who want to prevent HIV, there's still stigma around them. They don't want to be on a daily pill. Some of them that live with friends, family, roommates might not even know that they are on this medication. They don't want to be perceived as high risk or engaging in risky behavior or engaging in behavior that they might acquire HIV. So giving them a bottle that they need to take every single day for some of them is a concern. So some of them don't get on an oral PrEP because of this. They're scared someone in their household will find the medication. And then for people who travel, if we see a lot of instances where people travel and they forget their bottle. So now they're off their medication for 1 week, for 2 weeks, for 3 weeks. At that point, they don't have any protection against HIV. So with something like an injectable version of a HIV prevention medication where they get the injection for 6 months and they're really good to go. They can travel. They don't have to store their medication. You can't even store the medication. The medication will go to your clinician's office. You have to go into the office to actually receive the medication. You can't administer at home. So it's been very exciting to be a part of it. Patients are very excited. We've had patients who have been on an oral PrEP to prevent HIV for about a decade. Some of them are on it, had possibly been on this for years and years straight. Others kind of stop and restart depending on activity, depending on relationship status. But it's been nice to be able to give patients another way to reduce HIV, because HIV is a great burden throughout the country and really throughout the world.
Thank you. Thank you very much. Next, we're going to move to Oliver, who is going to begin, providing a bit more information on the access situation with lenacapavir right now. So far, we've heard from Dr. Wesley, who has discussed the early stage research, the importance of NIH publicly funded research in developing these innovations. We've heard from Saba about how these innovations really do impact patients, they expand options, and now we're going to move into the access considerations and what is happening currently in ensuring that people around the globe get access to lenacapavir. Over to you, Oliver.
Great. Thanks, Cassidy. Can everyone hear and see me okay? Yeah, okay. Sorry, I'm sorry for my gigantic head staring down at you, probably. So hi from Northern California, up here in I'm actually here in Pleasanton right now, my sister's house, dog sitting in the East Bay, but I live in San Jose. Thank you to UAIM and the organizers, and to Peter for the invitation to MSF to speak to y'all. And I'm really honored to be involved with this with Dr. Sundquist here and his, contributions to creating this life-saving drug and bringing it into existence, and to Ms. Sadat in terms of providing the clinical treatment to people on a daily basis. So thank you all. And thanks to all you attendees here too for your interest in access to medicines. It's so important, and to see so many of you interested in this is really inspiring. So really echo Dr. Sundquist in saying that. And shout out to UCLA, my alum. So go Bruins. So I've admired and actually followed UAM's work for a long time, working with MSF, Doctors Without Borders. I've worked on access to medicines work here with MSF for many years now. And so UAM and the work that they do all across this country and globally in terms of the university level for access to medicines advocacy is really, really, really admirable. So I just wanted to say that, and also that this area of lenacapavir access is actually one of my top priorities for work in terms of advocacy and the communications we do around it out of MSF. I don't think I need to explain to all of you what MSF does. I think you're all pretty familiar with Doctors Without Borders. We're an international medical humanitarian organization working globally. We've been working on treating HIV since pretty much the advent of ARVs, of antiretrovirals, including being involved in getting access to the first antiretrovirals when really it was only available in the global north, and through the Access Campaign, the former division of MSF. In the 90s, we were working on getting the price down and getting access to ARVs to our patients in throughout sub-Saharan Africa and other low and middle income countries, to the point now where HIV is a arguably a manageable chronic disease. And so medical innovation has always been something that we, as a medical organization, we follow and that we try to implement, because we want to use the best treatments, tests, vaccines, everything out there for our populations that we serve. And long-acting ARVs are part of that innovation. These new PrEP drugs. So right now we're using cabotegravir long-acting, which is given every 2 months. As an injection. We're using that in Eswatini, which has the highest HIV incidence in the world, Malawi, Mozambique, and Zimbabwe. And now, as Ms. Sadat really nicely summarized, lenacapavir is really this game changer of only 2 injections a year. You can see it as possibly, don't quote me, but as close as we have to an HIV vaccine right now, when you only have to get 2 jabs a year, and you can prevent HIV among vulnerable populations. So us wanting to get lenacapavir for use in our programs, we want to obviously purchase it at a fair price from Gilead. So we've contacted them multiple times, asking that if we could buy, we wanted to buy lenacapavir for our use and to start to roll out in some of our programs globally. But we were kind of getting ignored for a while, and finally we had a meeting with them back in February, where they basically told us they would not sell to us for a few reasons that we actually found kind of inadequate. One is they just referred us to the Global Fund, then go to them, get doses from them. The Global Fund only has a limited allocation of 2 million doses for the next 3 years, which is quite it's a drop in the bucket for in our view. It's probably less than 10% of the global need for what is seen for preventing HIV. And so they refer us to Global Fund and say Global Fund handles the allocation, go to them, which is a little disingenuous because Gilead's are the ones who gave them that cap supply. So for them to refer us over to Global Fund wasn't working for us, and we wanted to buy directly from Gilead and not have to use that capped allocation for other countries that are already allocated, but to get extra supply from Gilead and buy it from them. And Gilead themselves have said that they have the capacity to increase production for needs and in high income countries. And so if that capacity to produce is there, we'd like to tap into that and buy directly from Gilead. Second reason they gave us that why we could just wait until 2027 when some generics would come online. The problem with that is twofold. One is there are people now today who need this drug. They can't wait a year they shouldn't have to wait a year to try to prevent possibly being infected. So that's one thing. And the other thing is that this deal that Gilead has in producing generics with 2 generic companies starting in 2027 actually excludes a number of countries that we want to treat in. And some of those countries are actually countries where clinical trials happen for these drugs, middle-income countries, including Mexico, Peru, Brazil, Argentina, places like that. So while there are a good number of countries where those generics hopefully will be available, it's about 120, I think. There are still quite a few middle-income countries where there are key populations that need to be treated that may not be able to access those generics in 2027. If they are made in time. And just to note, who are those populations? They're the ones that we work in terms of MSF, whether it's conflict areas, active war zones, fragile health systems, as well as key populations that may be underserved, marginalized in different parts of the world, men who have sex with men, transgender individuals, sex workers. And so those are the populations that we want to try to get these drugs for. And the last reason they gave us was just kind of funny is like you said, these contracts, it's just a lot of work. It's too much work. And so that's to us not acceptable. So we decided to go public about this. So earlier this week on Monday, we sent out an open letter to Gilead, to the CEO, Daniel O'Day, coming from our director of MSF USA, Sharon Hassan, as well as our director of our South Africa Medical Unit, Tom Ellman, calling on Gilead to please sell this drug to us and setting a deadline of 2 weeks from Monday, so April 13th, to get back to us as to whether they will sell the drug to us or not. If not, why? What is your actual production capacity that you said is possible globally? And if yes, then great. And how quickly can you get to us and how much? Yeah, so I think I'll stop there. I'm at a little over my almost 8 minutes. So thanks. Sorry I talk so fast.
Thank you, Oliver. Now we're going to pass it off to Peter to further discuss these access considerations, what is being done, what's not being done, and what we can do about it.
Thanks, everyone. It's great to be back with you here. Thanks so much to Oliver and to Saba for your work. and Dr. Sundquist for this incredible contribution to humanity. It's really remarkable. Yeah.
So we're on the other side. Yeah.
So while they get that set up, so we all right? There you go. Sorry about that. You're good. You're good. Hey, everyone. You know me. I'm on the board of UAM, and I'm the Access to Medicines Director at Public Citizen in Washington, D.C. Thanks again for this opportunity, and thanks for this incredible breakthrough. So lenacapavir, it allows us to envision and even to map out an end to AIDS in our time. This is a disease that has killed hundreds of thousands of people or more, sometimes many more, every year of my life. I've lost some good friends, Matthew Blanchard, Javier Leo Varon. I've never known a world without fear of AIDS, but today it's possible. So thank you. Possible, but not without much more ambition. Do I click this here? There we go. Okay. So Oliver referenced this primary deal right now to get lenacapavir to low and middle income countries, capped at 2 million people for the next several years. I want to talk about 3 challenges to the kind of rollout that we need. These slides are going to focus on Gilead a bit, but we have more problems than that, right? So first one, of course, these extraordinary cuts that global health endured over the past year, they have eviscerated the architecture that provided life-saving treatment to people all around the world, the continuum of care, community organizations, clinics. Over the course of several decades, the world invested to help communities reach the nation state, reach the international community, reach the procurers and the distributors, to have a means of delivering life-saving care to people all around the world. And that has been devastated by the cuts to global health and dismantling of USAID. That is a major challenge that we are going to have to overcome in order to reap the promise of lenacapavir. Similarly, country ambition, procurement, and funding is far below what it needs to be today. And the world can't rely only on the United States, particularly in a time like this. We need to find a way for developing countries to allocate larger proportions of their budget. We need other rich countries to step in and take advantage of the situation that we have. But the third is this issue of timely, affordable supply. So we need many times more. And Dr. Sundquist, we were talking about this last night. You gave a figure. How much lenacapavir, I mean, what should be our target in terms of population at a minimum?
You want me to comment?
Yeah, so you were telling me, yeah.
So the people use number needed to treat, you need to treat about 50 people to prevent one transmission. And so that means that 1.3 million new transmissions, it has to scale up at least an order of magnitude, even to have 500,000 prevented transmissions. And that's an order of magnitude from the 2 million that Peter's talking about. So this really is a start, but it's only a drop in the bucket.
So you had mentioned, yeah, instead of 2 million, 20 million is be like the minimum to what we're getting. And so we're nowhere close. And we're starting to see some rollout. But for example, you see, we've got press releases from the State Department celebrating the delivery of the first, I think it was 500 or 1,000 doses to Southern African countries. And obviously that is not going to get us where we need to go. I find this statement with MSF kind of unfathomable, that Gilead would not sell to the frontline clinics that are making drugs available all over the world. You notice what they said. It's 2 million for low and middle income countries, but there's no indication at all that that's the limit of Gilead's supply. So what are we to infer from that? It doesn't make any sense, except that Gilead presumably wants to reserve the remainder of its supply to sell at higher prices to richer countries. And of course, that is not what the world needs right now. That means we're going to have to get tough with Gilead and its power. Go to the next slide here. Talked a little bit about regulatory delays and access. This is one of the problems too. So Gilead is rolling out in some countries, but most of the world doesn't even yet have lenacapavir registered. If the drug isn't registered, it's hard to make a national treatment plan. It's hard to say we're going to dedicate the following resources to purchase this drug. So 250 organizations have written to Gilead and say, at a minimum, you need to start registering your drug all over the world in all low and middle income countries, so that as one world, we can begin to make a plan for how we're going to ensure access to this drug. There's sort of a circular problem of ambition. We want countries to step up and make financial commitments, but they don't know when the drug is even going to be available for sale in their country. So they want Gilead to register it, but Gilead wants the financial commitment. We think Gilead needs to be a first mover here, as well as, and to register further. Also, you can't bring in generics in the market, for the most part, if you don't yet have an originator registering the drug. So for pricing, Gilead, we only have an estimated price from Gilead, because one of the problems, of course, we have is transparency. Gilead hasn't actually told us what their pricing is or will be. But the best information seems to be a price of about $100 per person per year through this global fund deal to buy Lenacapavir for developing country use. But researchers think it can be produced for quite a bit cheaper at a profit. And you say, so these prices are 35 to 46 or $25 at higher volumes. That includes a reasonable return for anyone manufacturing the drug. So we think the price can come down quite a bit from where it is today. Then there's the problem that Oliver referenced of licensing arrangements, something UAIM works quite a bit on and something I work quite a bit on. No, I mean, it can be said, right? Gilead didn't have to license, and it's important that they brought in 6 generics manufacturers to help supply the world with this drug starting next year. So there are a couple different ways of looking at it. One is like Gilead took that step. They were pushed very hard to do it. However, as Oliver said, they left a lot of the world out. And Gilead now has a financial relationship with the 6 most likely competitors, right? So instead of potentially competing with Gilead, these organizations will now work with Gilead. So another way of seeing the licenses is that Gilead has sort of carved up the map, and it is ensured its control over where Lenacapavir will be distributed. There are still some things we can do about that. People like me work on compulsory licensing, the option of the countries that are left out of this arrangement to seek other generic suppliers and try and trigger generic competition outside the license. And we're hopeful, but it is tough when you have some of the most likely producers already bought in to an arrangement. They're not going to supply outside their deal with Gilead. So we have to find new potential providers in order to meet the need. Otherwise, everyone will be just subject to Gilead's decisions about what the price will be and when the rollout will be. We talked a little bit about the high-income countries listening to Saba, but of course the price here is exceptional. And even if insurance covers, that's a pretty significant burden on our healthcare systems. Some of you may have noticed that right now we're having an AIDS drug crisis in the United States, not on Lenacapavir, but on actually related to other Gilead drugs in our AIDS drug assistance programs, where people are getting kicked off the rolls for these assistance programs because they're no longer solvent. They don't actually have the money to pay for Gilead's other high-priced drugs. So price is a pretty significant problem in the United States. Florida just passed an emergency measure to prevent 16,000 people from getting kicked off their ability to access AIDS drugs here. So, and just like a note for context, I guess, as we're sort of thinking about the relationship, we can distinguish, between the research that companies do and what we want to do after a drug comes to market. Gilead made $30 billion last year and projects $800 million in Lenacapavir revenue just for PrEP alone this year. So it's pretty lucrative for them, and it's hard to think of why they can't sell to Doctors Without Borders. Anyhow, so what can we do? Again, with these 3 prongs, right? You got the problem of Gilead and pricing and patents, but we also have the problem of country will and health infrastructure. For Gilead, there's U.S. price reform, including expanding U.S. drug price negotiations, significant victory that we won legislatively in recent years. We can challenge the patent power. This is the thing that pharmaceutical companies are built around, the lucrative patent monopoly. And when we challenge that power through a mechanism like compulsory licensing or through the work of a group like UAIM, they actually become much more willing to sit down and negotiate better prices. That is our leverage. Our ability to sort of go after the patent monopoly is our leverage. And we need to raise expectations for Gilead on registration and supply. A challenging thing about Gilead is that it has very close relationships with the HIV community, and for good reason, right? It's developed a number of life-saving HIV drugs, but it can't be the case that we just say that's good enough when only 18 countries initially are being contemplated for the first rollout of lenacapavir, when Gilead won't sell to Doctors Without Borders, when there's only 2 million doses being made available. We need at least 20 million in order to deliver on the promise of this drug. We simply have to raise our expectations on a company that pulled in $30 billion last year, right? We have to be able to say, that's great, but we demand more. But secondly, we very much do need countries to step up. We were talking on the panel earlier today, we had a question about, well, we're not in the United States. What do we do? Canada, somewhere else. We very much need those countries to step in and start to fill the gap and start making commitments to match. And we have to do the painstaking work, third, of protecting and rebuilding health all the way down to the community level. It's not going to be an easy project. We're not, USAID is gone, right? It's not coming back. But there are projects and conversations underway for how we start to rebuild some of this programming over a period of years. And even inside the Trump administration at the State Department, for example, there are people who are now negotiating new health agreements with many countries. And, unfortunately, those agreements are also secret. I'm happy to announce that Public Citizen sued yesterday to get access to the text of those agreements so we can start to understand what is the money that is being reinvested, how is it going to be accountable, where are the investments in HIV, what are the pledges as regards lenacapavir. But it's going to take time, right? And I think for all of us in this room who are dedicating our careers to science and health, we're going to spend this is going to be a decades-long project of how we together figure out to create an infrastructure that can successfully deliver health again to everyone who needs it. And clearly none of us can do this alone, but we in this room have essential contributions to make. So I wanted to wrap up with access planning, because it's also interesting to reflect on UAIM's approach and Dr. Sundquist's problem, how to get at these technologies early on. We spoke on the phone the other day, and I think you don't mind me saying that there was, one of the dilemmas here in the early research was that the University of Utah didn't actually have intellectual property in the invention, and so couldn't just write terms into the license. But UAIM is now working on access planning provisions that go further and further upstream, and we can try and imagine and work with researchers, maybe discuss in the Q&A a little bit, how do we start normalizing access so that it's there right from the beginning of the bargain, right? It's not only a consideration of how does the drug get to market, but we have a responsibility to the world. We can foresee potential applications, and even in something like a non-disclosure agreement or early conversations, it doesn't have to be very specific. It can be a plan to make a plan, but that we will have expectations about access down the line. Just since we're here in plenary, I think it's worth saying a word about how much progress this access planning is making right now. So here at UCLA, you successfully were able to get your administration to adopt terms that will require drug makers to provide access conditions on technologies that are developed and funded here. And we talked earlier about a California bond initiative to achieve the same statewide. You're very far from alone in this. We saw that once a drug is on market, it's a street fight, right? To try and get Gilead to reduce prices, to try and negotiate better prices, to try and fight over the patents is actually very difficult. And people on the other side of the table for me have some point when they say, well, we don't want to just like take this stuff away, take the patent away. There's investors, there's expectations of profit. And the advantage of what UAIM is doing now is that it's right there at early development and everyone's on notice. And if everyone starts to adopt these sort of conditions attached to research and development funding, the public funding that we put forward, the public inventions that are used, then it can become sort of an industry norm that everyone understands. Like we are going to have time to plan for how we are going to ensure access in the end stage. So it's seen as more fair. And in recent years, in addition to UCLA and UC Berkeley, the nations of the world, through the World Health Organization, adopted a pandemic treaty that includes these same principles that UAIM has been advancing. And the National Institutes of Health adopted access and equity planning so that, for example, had this been active in 2020, the NIH Moderna vaccine, the world's most effective COVID vaccine, probably would have been available to many more people early on at a more affordable price, because the US government would have had the leverage to insist on those terms and conditions. So the National Institutes of Health, the world's leading biomedical research funder, now has access planning as well, due in considerable part to the example that people in this room have set. And it's a bipartisan policy. It was incubated under the Biden administration and approved again by the Trump administration, the Office of Management and Budget. So this is surviving and moving forward, and it's something that we can build on. In any case, this clearly, it's all of our responsibility, quite possible to make it better, to change lives, eventually even end AIDS. And we know that because we've come so far together already. So thank you. Thanks all so much for your work. Thank you, Dr. Sundquist.
Thank you, Peter. We are going to get Oliver back up on the screen. I'll invite Peter to come sit down at the table, and we will start our panel discussion. We really want this to be interactive, and so get your questions ready. We'll start off with one question from me, and then we'll move into about 30 minutes where you all can ask all the questions you would like. But thank you so much to everyone for starting off with some intro remarks. I hope that told the story of Lenacapavir as this really amazing research that started in a university lab and is now possibly going to change the course of the HIV epidemic. So just to start off, I do want to really nail into what Peter said there at the end, talking about access and embedding access really at the beginning of drug development. But I know Dr. Sundquist would probably say that's challenging, because I don't know if you noticed as he was going through his slides, a lot of those citations at the bottom had years that started with 1990 something. And so this has been a very long time coming. So I think it is really interesting to see just the scale of time that it takes for a drug to start from early stage discovery to actually something that can reach patients. So I would love to hear, I guess, first from Dr. Sundquist, and then as others have perspective as well. First of all, the role of public funding in these really instrumental discoveries that are going to change the course of an entire disease. And how do we as advocates think through what's the role, first of all, of public funding? Would this have been possible, those early stage discoveries, without public funding? And then kind of zooming out, and this is maybe something Peter can talk about, is how do we find opportunities to impact that, especially when some of these early stage discoveries happened 25 years before the medicine is actually developed. I believe this mic, you might have to switch it on. Perfect.
Okay, so thank you, Cassidy, and thanks to the other panelists. Those were great remarks that raised important issues. So let me start with the one that I think is easy. I think that I tried to make the point that basic research is a long-term process, but that I think we can promise as a community that we will make discoveries that transform the way medicine is practiced, even though we can't predict exactly what they will be, because they almost always come from unexpected places, because transformational discoveries are, you weren't thinking about something one way and now you're thinking about it a different way. And that means that it's not engineering, it's unexpected discoveries. But I think if you look at the history, what you find is that this happens time and time again. And so even though individual labs and discoveries may only add to knowledge, which is an important thing to do, a few of them will change the way we do gene therapy and so forth. So I think if we don't fund NIH well, what will happen is in the short term, we won't feel it that much because these are long-term problems. But what we'll discover is that our feedstocks of brand new transformative ideas have gone away. And so, right now I'm a department chair. We have 4 really talented young scientists, young faculty who have their own labs. They have good NIH scores and they don't yet have their grants. So this is really affecting people. I'm peripherally part of an HIV vaccine center. So we'd still love to have a vaccine. A year is a great thing. The next Gilead version is going to be a year long, but it's not the same as lifetime protection. And so even though we haven't yet had catastrophic problems, we have real problems through the current policies. So that's, and then so advocacy for NIH research is something I feel passionately about, and on the basic as well as the translational spectrum. I mean, we heard how important it is to have it across the, clinical delivery. Every one of these things matters. The more challenging problem, which I actually think about a lot, is when we were working on the capsid, we much more felt like nobody's interested in this as a drug target than we felt like people were beating down the doors. And so eventually we talked to a lot of drug companies, and eventually Gilead and Boehringer Ingelheim started capsid inhibitor programs. Boehringer stopped because they weren't successful, and that's something we have to acknowledge happens a fair amount in the drug industry. And Gilead succeeded spectacularly. And as you heard from Peter, we didn't really have intellectual property because all we'd done was tried to understand how does the capsid function and why is it important. And so we published everything in the public domain. Anyone could use it. But that meant that we didn't have so much IP. We didn't have any IP in this particular case. So one thing we're trying to do in our department, we now have a translational advancement fund where we're taking discoveries in the labs and funding them for a couple of years to make them more translational. And we're doing that, so that means maybe having an inhibitor. So if, for example, we had a lead inhibitor, which we didn't, that would have been much more attractive to drug companies. And that's an important issue. Getting them interested in the first place is harder than you think it is. But also that would have given us much more leverage. And so we're trying to do that within our department because I think that will change where we start in the process. So I guess those are some comments. I have to say that our relationship with Gilead, the scientists, has been spectacular. One of the people I work with, I have the greatest admiration for. I have no intellectual stake in Lenacapavir. My boss, the dean, asks me why Utah isn't going to get a lot of money. They're not. But I feel like when I think about where we were, I think we did the only things that were really possible. And I think if we hadn't taken that path, there probably wouldn't be a Lenacapavir, but others can disagree.
Yeah, we have a collective action problem, right? Because the major funders for research and development are concerned that if they ask too much, then the drug makers may move on to a different source of technology or funding. But the really excellent news, and UAIM has made a major contribution to this, is that's changed in recent years. And we earned the change in lives, I'm sorry to say, because it's partly a product of the terrible experience of COVID inequity. And so many millions of people dying for lack of vaccine access moved a lot of health organizations to get a little more serious about what kind of access conditions they ask for. And we've all pushed hard toward this end. But so now there's a conversation being driven by many of us sort of all over the world. The Coalition for Epidemic Preparedness Innovation, CEPI, which funded many vaccines developed in COVID-19, has raised its standards. We've all pushed them to raise its standards, and it's actually advocating to other funders and saying, governments, NIH, Japan, Wellcome Trust, Gates, please start thinking about access planning. When you give out grants, think about how you're going to build access conditions there in the beginning. And as I said, you see it now in the pandemic agreement and out at NIH. So the work that we do in this room, I actually see this as like the central value proposition of UAIM for me, at least in terms of like our strategic objectives over the next couple of years, is this: how many universities can we bring along to adopt only what we've already done at UCLA, UC Berkeley, and NIH and just see that make it an inevitable tide. If everyone who is funding medical research and development has some basic expectation that there will be at least a discussion about an access plan, soon that will be the norm. And from there, we can raise the floor of what that means, go from more vague commitments to more specific commitments, and continue to raise standards over time. So I think that's the big project. And, traditionally in this organization, we've used the link of like, oh, well, the university has a patent, we can license the condition. But when you're talking about something as flexible as access planning, the commitments can be, and the leverage can be a lot of different things. It can be a plan to make a plan. It can be, a value statement initially. The lawyers we talk with about this say, the earlier in the R&D you go, the more flexible you're going to have to be, the less specific the request. But this is what we're trying to normalize, that the investment, I think here's the principle. The investment is not only for the innovation, the investment is for the access, right? We are asking for innovation and access at the same time. And of course, that should be the basic deal in the public funding of any biomedical research, or the concession of a license to a federal invention like the key inventions behind the COVID vaccines, which were designed by federal scientists. When we make that progress, when we enter into partnerships that are going to be lucrative in the end for someone, the innovation and the access together is just what is the public deal that we should expect. Unfortunately, I think this is one of the areas where we are making very significant progress today. And I think if we make some smart plans together, like, we're not going to be alone, right? I mean, it's seen as, this way of thinking about access is seen as more acceptable to people that we sometimes fight with, because there's far advanced notice, businesses can plan, there's flexibility, can be designed a lot of different ways. So I think we're on the verge of something very meaningful here that it could save a lot of lives over time.
Oliver, do you have anything to add?
All right, trying to find my unmute button. No, I think that's what was said by Dr. Sundquist and Peter was spot on. But just to echo what Peter was saying about innovation plus access, that really, I think, needs to be the norm now, as opposed to just, we create these new breakthroughs and then it gets privatized and monetized. So that plus access part really needs to go hand in hand with R&D and innovation, especially when it comes to health.
And just to kind of piggyback off of that, and before we open up to questions, I would love to hear a bit more from Saba and also Oliver. What's at stake here? What happens if we don't scale up access to patients either in the US or to MSF's patients abroad? What are the two worlds that we could live in? One with global access to lenacapavir, and then the other where, just the wealthy are able to access it.
I forgot to mention when I was up there, but people who are at risk for HIV acquisition, there's a large intersect between substance use disorder, homelessness, mental health disorders. So we are really trying to reach the most vulnerable populations out there. And then some of them have a hard time even getting to a clinic every 2 months for a daily pill. So something like this, where it's 6 months and they're good to go for 6 months and they can come back a little bit later than with some of the other options out there, we're hoping will really bring down the numbers for HIV. And for some people, there are dreams of HIV eradication. I don't know if it'll be in my lifetime, but that would eventually be the goal.
Thank you. And Oliver?
Yeah, I mean, there's real parallels to what Ms. Sadat just said with what we see in terms of the countries that we work in globally. It's the same thing in terms of people not being vulnerable populations, not being reached, underserved populations, still not being able to get these preventative treatments, and then you see the HIV incidence spiking again. This is why when we work on HIV, when we're doing treatment, and we still do, but when when we look at treatment, we use with those countries that have had the highest levels of HIV prevalence and incidence, right? So we were going to those places where the needs are the highest medically, and providing treatment. And now when we have these preventative tools, we do the same. Eswatini being an example of the highest HIV incidence globally. Countries like that, if they're not able to get the tools that they need in terms of prevention, then yeah, you will see these populations continuing to get HIV at a high level, and then it kind of snowballs. So very similar to what we see at a local level with Misarat and what we see globally in low and middle income countries.
We will open up the floor to questions. Does someone want to kick us off?
So thank you all for the panel talk. One question I have is regarding the Global Fund partnership, which they had with Guinea, I believe. My question is why, even though there's a partnership with the CEO, why are there only 18 priority countries that are targeted? And specifically also, why is there like a specific number allocation of 2 million doses given? Is it both, from what I understood, was it because the data they were looking was outdated? And yeah, because I understand that also Lenacapavir is an amazing drug, but also I understand that viruses they also mutate eventually, and Lenacapavir in the future might not even be as effective if we just delay not giving the medication to everyone, like the global south, who won't necessarily have access to these drugs. So my question is really why is there only 18 priority countries targeted, even though there's a partnership with the Global Fund, which I believe the mission is to eliminate the diseases globally.
Well, I'm not privy to what their internal discussions were, but Gilead has said that those are, sales at a not-for-profit price for them, and the inference is that that's as much as they were willing to accept, I think, and the most also that between the price negotiation between Gilead and the Global Fund, how much the Global Fund could afford, how low Gilead was willing to let the price go. That was sort of like the high threshold figure, like that's the biggest number we can get to. And presumably they want, collectively a big impactful figure, but Gilead's not willing to let more of the market go than that, and Global Fund having trouble raising more money than that. So I think that's sort of the overall target. And 18 countries, there are other knowledgeable folks in this room who may have a little more insight into this, but it's high HIV incidence countries where there's a history of partnership. I mean, they're priority countries, right? I mean, it makes sense. But what doesn't make sense, what we haven't had a good explanation for, is why not at least start registering the drug in many more countries? It doesn't seem like to us like that can be that expensive or difficult, but that is indeed where we're at. There's progress, but it's slow.
I hope I've made it clear that I'm not a spokesperson for Gilead positively or negatively, but one thing a distinction is worth making. So you've heard that they have negotiated with 6 drug companies that they will give license-free, royalty-free permission for those 6 companies to distribute drug at a small profit, but at a much lower cost. And that's 120 countries. So I think it's absolutely valid to ask, why do we have to wait and so forth? But just to be clear about where it's going to be available, the 6 companies that are making generic drug will have 120 countries. And then I think if Gilead were here, and I'm channeling my inner Gilead, I'm not them, that they would say that they also envision tiered pricing so that middle income countries won't pay the same as we do in the US. That I think everybody's asking good questions. We want to, these are real individuals who are really going to get HIV. And even from an economic point of view, the economics say it costs you 7 times more to treat a person for life with HIV than it does to prevent them from getting it. And that's, of course, not talking about the human suffering that is associated with being HIV positive, especially in developing countries. So these are important points.
Wonderful. Do either Saba or Oliver want to comment? Okay, perfect. Other questions?
Hi, I just had a general question for my curiosity. Maybe Saba, you can answer this. So when people really understand their health risks, they're more likely to engage proactively by adopting these preventative measures. Has there been an increase in HIV or just STD pathology literacy that will further augment usage of preventative measures like lenacapavir?
So yes, because if patients are not aware of these options, then they're not going to be able to access them. So in general, any sort of STI education will help with prevention, but unfortunately, that's not very great. I think in this country, especially in certain parts of the country more than others. So that's what we try to do at our clinic is we really try to provide STI education. The problem is a lot of these STIs, HIV are a little historically have been more associated with LGBT communities. So that's why a lot of people might not be comfortable discussing them in spaces that they might not feel are LGBTQ friendly. So that's something we have ran into our clinic. So people will come to our clinics because it's specifically HIV, STI for those sort of treatments, education, prevention, instead of going to primary care where they might not even feel comfortable sharing their status or information about their activity. But really, any sort of information about STI prevention will benefit prevention, and then something like PrEP is great. So even for people out in the audience who might have never heard of PrEP, I'm pretty sure UCLA Health does have oral PrEP available if you are insured through UCLA. And if you've never heard of it and you think this might be great fit for you, then I would say try it and see if it is.
Thank you. There are other questions?
Hi, and thank you guys again for an amazing panel. Something that I am curious about, obviously, there's been like a lot of talk about like preventative measures that we take at UAM and how to make sure that like before privatization, we put in like access planning. But when it comes to something like this, where it's a more complex situation that's already like post-market, what can students and people in our position do to like advocate for like legislative changes or anything that might be able to like improve the affordability or access around drugs like this?
Yeah, so I think we have to raise expectations is the number one thing, right? I mean, this is it's a transformative technology, but only to the extent that we can deliver on its promise by radically expanding its rollout. And that's a message that the world should hear, even in this time of scarcity. You know, we're very far from alone. I mean, again, despite the cuts, despite, you know, the fights and sort of different views we have about how technology should be handled, everyone from the Trump administration backs this technology too, and they very much see it as sort of doing well by doing good to facilitate its rollout, because it's an American company, right? The American company also benefits as a result of the U.S. government buying the product and trying to roll it out to the world. So, but the question is, you know, are we going to accept this kind of relatively low floor that's been established relative to the need, or do we raise our voices collectively and say this is an historic moment that we can't afford to allow pass us by? And there, of course, students have a key role. It's simply by being louder about it and by and by talking with people, raising voices, writing, we contribute to that clamor. There are, of course, legislative fixes too, or, sort of different policy fixes too in different countries. It gets pretty specific pretty quickly, who can work on what piece where. We for our part, we're actually hoping that we can trigger more substantial generic competition for the places where it's not scheduled to reach by working with governments that are not in the licensed territory and may be able to work on that. And if we're actually able to trigger something, that's a policy that will need support in public defense. So we're also working on, many of us sent letters to Gilead saying, publish your prices, show us the registration plan. And we can make the same asks of our governments too, though, right? Or is to sort of step up the spending in this area. There are many aspects to this particular challenge. We've got to rebuild the health systems. We've got to get the funding. But I think just indicating as much as we can that we have a real opportunity that we can't afford to miss is sort of the key thing to contribute for Lenacapavir at the moment, for the fight against AIDS.
Oliver?
Yeah, this is a great question, and thanks for bringing it up. And this is actually why we decided to go public now with this open letter to Gilead, and we put out a press release, and we've gotten some great press out of it and some real media attention. And it's something that we're looking to now sustain throughout the rest of the year of having this more of a public-facing campaign to be able to increase access to Lenacapavir for the global South. So that's when we start launching that campaign on a larger scale, this is exactly where we would need the support of groups like UAIM and student groups and Friends of MSF, which is a university chapter network that we have through Doctors Without Borders, and that's when we can really look to you all to amplify our messaging and amplify that advocacy to at some level increase the access to Lenacapavir. And just to make a point of what it's also what Peter just said, it's not everything we do is not just public campaigning. We also do a lot of bilateral policy advocacy. We have people who talk directly with governments and try to also work behind the scenes, behind closed doors. But when that fails or we need to go public, then we do things like put out this open letter and a press release and get this public attention, which is exactly where we're going to need you all here in the room to support us on. So stay tuned for that.
Thank you. And we will take one final question and then we will wrap up.
Thanks. Hi there. This just makes me think, how do we hold Gilead accountable? And actually, they're accountable to their shareholders and really no one else. And, with the state of the federal government here in this country, just constantly reminds me that we need to usher in a new progressive era in this country, that, corporations are people here. But I think the people need to take back the power, and that's what we're really talking about here. So it's not really a question so much as a statement. And yes, sorry.
Just to quickly address something you just said there, one of the things that we're looking at doing is providing this our advocacy here and our messaging directly to Gilead shareholders. Their next shareholders meeting is on April 30th at the end of this month. And so we're going to actually reaching out to them and other investor groups being like, if you invest in this company and this company is not selling to us, it's just something that we want to flag for you and for you to maybe ask your company about and that you've invested in. So that is actually one other avenue we're looking at is going that investor shareholder route and talking to them directly and saying, listen, your company's not selling. Can you ask them why?
Thank you. Could you all please give a round of applause for our panelists? Thank you all so much for joining us, everyone. Oh, Saba, do you have?
I have one final remark. Yep. By no means do I think PrEP is for everyone. I'm not trying to push PrEP. I don't get paid by PrEP. It's just, I just mentioned that for people who might have never heard of it or who were concerned, but everything you have to weigh the risk versus the benefits. And some people, their risk or their exposure rate might be higher than for others. So it's not for everyone, but if you find yourself in certain categories and you want to explore PrEP, there are options out to prevent HIV.
Thank you.